Okay, so now let's take a look at some practice problems here. And these are going to be related to what we were just talking about. Let's say we crossed a pure breeding tall and round seed pea plant, which is what we had just done above, with a pure breeding short and wrinkled pea plant. Okay, so you should probably be able to do this. What would you expect the results of this cross to be, assuming the traits, and I had to change because I made a typo, the traits of height, because we're talking about tall or short, and seed shape, because we're talking about round or wrinkled, we're on different chromosomes, which is what they are. So this is kind of what we've done previous to this week. So the results of this cross would just be that this is giving the T, capital R alleles, and this is giving the lowercase t and the lowercase r alleles. And so we're going to end up with hybrid offspring. This is in the F1 generation, or first filial generation. And then what would we expect as a result of a back cross of an F1 plant to one of the parent plants, which is this one? Okay, so again, that parent plant is again just going to give the little t, little r, but a hybrid is going to give, we'll do the FOIL method, that, or that, or that, or that. So I'm going to just do, I'm not going to do a giant 16 grid Punnett square. I'm going to do kind of a 4 grid row, because I can just put this one here, and then I can put my first, outer, inner, last FOIL method just right here, and then I can fill this out. So I'm going to get 25% of them looking like tall and round. I'll get 25% tall and wrinkled seed. I'll get 25% short and round seed. And I'll get 25% short and wrinkled seed. Okay, so that's if you know, they were normally on different chromosomes and they are. Okay, what if they were on the same, hypothetically, if they were on the same chromosome, what would the results look like? Well, it would be different. I would still have one of the parents just making these little t, little r gametes. Okay, so that's for sure going to happen for one of them. So we're still going to just set up our grid with little t, little r on that side. Okay, but remember, if I have this hybrid here, okay, that on a cellular level, if the parents of this were this and that, and so one parent gave a chromosome that had a capital T and a capital R on the same, stuck together, right? There's the chromosome, here's the T, here's the R. And another parent gave a T and an R on another chromosome that on a cellular level here, if I were to look at this cell, it would look like this, T, R, T, R. So even though these look like they're stuck, they're like all messed up, they're not. These two are on that chromosome, and these two are on that chromosome. Okay, so that's what we have to consider, knowing the lineage here. And so these, unless you have crossing over, of course, these are not going to mix with those. And then, you know, when this cell goes through S phase of interphase, we're going to go just like we did before with T, T, R, R capitals on that side. And here we have little lower t's here and lower r's there. And this cell is going to split in half, and we're going to end up, right, with these kind of being split up, and we're going to end up eventually with 4 gametes looking like this. And those are the gametes that I'm going to put on my cross here. So I'm ending up with 50% looking like either of the parents, right? So these look like the hybrid parent, And these look like the little t, little r parent. And I don't get those, let me highlight these here. I don't get those intermediate phenotypes if they're on the same chromosome. Well, I do. Remember, I do get those a little bit only as a result of crossing over. So if, I didn't show it here, but if, you know, these end up like crossing over and we end up getting these chromosomes to kind of switch out some of these before they split up and such, then we're going to end up with some of those intermediate. So you might get, again, like a smaller percent. You might get like, 40% looking like that, 40% looking like that. But then you might still get some of these other ones where you're like big T, little r. You might get like 10% of those. Or little t, big R, little r. You might get like 10% of those, again, as a result of crossing over. Those are called recombinant phenotypes, okay? Because they have to have been the result of crossing over events to occur if these genes are on the same chromosome. So those, again, are called recombinant phenotypes. Number 6, looking at fruit flies, because fruit flies tend to reproduce pretty quickly, and there's a lot of traits that you can look at, and so it's pretty typical in laboratories that are studying genetics. I know I looked at fruit flies in college and stuff. Fruit fly experiments, we would run these. And it's interesting because a lot of the traits are actually on the same gene. And so you can start to look at this type of stuff in real life. So here's an example question on fruit flies. I kind of modified a type of question that you would see in AP biology just to kind of go over the concept here. So to start with, let's just call this the wild type fruit fly. A wild type means that it's what you would normally see in nature, in the wild. And wild type also will denote what you would call the dominant phenotypes. Okay, so it's dominance versus recessive. And so the mutant here is going to show some recessive phenotypes. So as you can see, wild type has maybe a tan coloration, brown coloration, and the mutant has a different coloration, which would be recessive. The letter E is used here. It's called ebony color, so that's the use of the letter E. And then the wings, VG, stands for the fact that these are vestigial wings, or not really fully formed wings. So in a parent generation, if we cross a wild type homozygous dominant with a mutant homozygous recessive, we'll end up with a hybrid, which has both the recessive and the dominant alleles, but shows the dominant phenotype because it has at least one dominant allele. But if we take females from the F1 generation, this is the F1 generation. If we take females from the F1 generation, so heterozygous or hybrids, and we cross them with true-breeding male flies with the recessive. So we're going to cross, we got a lot of scribbles up here. We're going to cross this genotype with this genotype, okay? Now, what would happen in this situation, if the genes for body color and wing shape are on different chromosomes? Okay, so that would be like, again, the normal situation. So if the genes were on different chromosomes, since I know that the mutant or whatever can only provide the lowercase e and the VG genes, I can just put this here. And I don't have to have a giant 16 square, Punnett square. But with the hybrid, we have to do the FOIL method now. So we have to go first, outer, inner, last. And that's kind of confusing because we're using some letters that we're not really used to using here. So I'm going to go E, VG. And if I did this, I'll just write it out here. This will look like the mutant parent. Okay, that's going to look just like this one. Okay, let's do another one. I guess I'll just look at this one here. This one would be next, E and plus. And I'm going to write VG plus so I know what I'm talking about. So this has a dominant allele. So this would be ebony color and normal wings. And this was ebony and vestigial wings. Okay. And then if we do this one next, that's E plus VG. So this one is tan color and vestigial wings. And if we do this last one here, we get tan color and normal wings. That is, again, so that'd be 25% chance. And this one looks like the normal parent, or the wild, I don't know, like normal. Looks like the wild type parent. That one looks like that one. And those look like it, and then this one looks like this one. So you get 25% looking like each of the different parents, but you also get these recombinant phenotypes if they're on different chromosomes. But remember, if they're on the same chromosome, I'm not going to run the whole cross again. If they're on the same chromosome, to answer this question, then we would end up with like more of these and more of these, because they look like, each look like one of the parents, and we'll get less of these, because the only way you're going to get those and less of these, the only way you're going to get those is if you get recombination or crossing over and new combinations being formed. To give example of how many of each offspring of each phenotype could be expected, again, I'll just say like 40, and it depends on how close they are, 40 of these, 40 of these, 10 of these, and 10 of these, or something like that. And again, depending on how close to each other these traits are, these genes are.